Cross-region neuron co-firing mediated by ripple oscillations supports distributed working memory representations. The core question. The paper addresses a fundamental puzzle in human neuroscience: how do distant brain regions coordinate their activity to support complex cognitive functions like working memory? While high-frequency oscillations have been proposed as a mechanism for neural integration, it remained unclear whether these oscillations actually facilitate communication across large distances during active cognition, and whether this coordination carries behaviorally relevant information. What the researchers did. The researchers analyzed an open-access dataset of intracranial recordings from 35 patients, 43 sessions undergoing diagnostic monitoring for epilepsy. These patients were implanted with microwire electrodes targeting 5 brain regions bilaterally: the hippocampus, amygdala, ventromedial prefrontal cortex, anterior cingulate cortex, and pre-supplementary motor area. Patients performed a modified Sternberg working memory task. In each trial, they viewed either 1 or 3 images encoding phase, maintained those images in memory over a delay maintenance phase, and then identified whether a probe image matched the original set retrieval phase. The task allowed the researchers to compare neural activity across different memory loads and across fast versus slow reaction times. From the recordings, the researchers isolated 1,373 single units individual neurons and detected ripple oscillations, brief 50 to 100 millisecond bursts of high-frequency activity centered around 90 Hz in all 5 brain regions. Ripple characteristics were consistent across regions: densities around 0.5 ripples per second, amplitudes ranging from roughly 6 to 10 microvolts, and durations around 70 milliseconds. Finding 1. Ripples co-occur without distance decay. The researchers first asked whether ripples in different brain regions occur at the same time, what they call co-ripples. They found that within the same microwire bundle separations under 5 mm, the median co-occurrence rate was 13%. Between bundles within the same hemisphere separations of 71 to 203 mm, the rate dropped to 5%. Surprisingly, across hemispheres 35 to 223 mm, the co-occurrence rate remained almost identical at about 5%. In other words, once ripples extend beyond the local bundle, their probability of co-occurring with another region stays relatively constant regardless of distance. This lack of decrement suggests that co-ripples reflect a brain-wide network phenomenon rather than simple point-to-point transmission. Finding 2. Co-ripples enhance cross-region co-firing. To determine whether co-ripples actually facilitate neural communication, the researchers analyzed co-firing defined as spikes from 2 neurons occurring within a 25 millisecond window. They compared co-firing rates during co-ripple periods versus no-ripple periods. Across all 31,489 unit pairs analyzed, co-firing rates were significantly higher during co-ripples, with a median increase of 34% compared to no-ripple periods. This enhancement was observed across all connection types: amygdala, cortex, hippocampus, cortex, amygdala, hippocampus, ipsilateral cortico-cortical and contralateral cortico-cortical pairs. The magnitude of enhancement was broadly similar across these pathways. Critically, the researchers performed rate-corrected analyses to ensure that this effect was not merely due to both neurons firing more independently during co-ripples. They used the spike time tiling coefficient STTC, which explicitly corrects for firing rate. STTC was significantly above 0 during both conditions, but was 117% greater during co-ripples than no-ripple periods. This confirms that co-ripples enhance genuine temporal coordination, not just elevated excitability. Finding 3. Ripple and unit rates increase during task phases. The researchers examined how ripple rates and neuronal firing rates varied across task phases. During encoding and maintenance, ripple rates increased moderately compared to baseline in all regions, roughly 10 to 13% increases. The retrieval phase showed the most pronounced increases, particularly in the pre-supplementary motor area, 36% increase, and hippocampus 13% increase. Unit firing rates showed similar but not identical modulation patterns, with modest positive correlations with ripple rates across all task phases. When comparing high memory load 3 items to low memory load 1 item, co-ripple rates increased significantly during maintenance and retrieval. During maintenance, the increase was greatest between the anterior cingulate cortex and pre-supplementary motor area, and between the anterior cingulate cortex and hippocampus. During retrieval, the greatest load-dependent increase was between the hippocampus and pre-supplementary motor area, and between the ventromedial prefrontal cortex and hippocampus. Finding 4, ripple activity predicts behavioral speed. The researchers also examined whether ripple activity was related to reaction time. For high-load trials, they compared ripple rates during fast responses below median reaction time versus slow responses above median. In the hippocampus and amygdala, ripple rates were significantly higher in the 300 to 600 millisecond window after the retrieval stimulus for fast responses compared to slow responses. This association suggests that ripple engagement predicts retrieval efficiency. Temporal analysis revealed that hippocampal ripple onsets consistently preceded amygdala onsets, indicating a temporal sequence in limbic structures during memory retrieval. Finding 5, specificity to the ripple frequency band. To determine whether these effects were specific to ripples, the researchers performed parallel analyses on oscillations in the low gamma band 30 to 55 Hz and non-oscillatory very high gamma activity 120 to 190 Hz. While these other signals also showed task-related increases, the load modulation was substantially weaker. During retrieval, linear mixed effects modeling revealed significant load-dependent modulation across 12 of 15 region pairs for ripples, but only 2 pairs for low gamma and 0 pairs for very high gamma. This frequency specificity indicates that ripples occupy a unique role in long-range coordination. Finding 6, co-ripples drive task phase modulation of co-firing. The researchers then asked whether cross-regional unit co-firing changes across task stages, and whether this depends on co-ripples. During co-ripple periods, cross-regional co-firing increased significantly from baseline in all task phases, 28% during encoding, 29% during maintenance, and 24% during retrieval. In contrast, no-ripple periods showed minimal changes, 1% increase during encoding, 3% decrease during maintenance, and 4% decrease during retrieval. When looking at all periods combined, the increases were modest, 9% during encoding, 4% during maintenance, and 2% during retrieval, driven largely by co-ripple contributions. Furthermore, memory load modulated this effect. During co-ripple periods, co-firing was substantially higher in high-load versus low-load trials, 13% higher during maintenance and 19% higher during retrieval. No ripple periods showed negligible load modulation. Finding 7, co-ripples reinstate stimulus-specific firing patterns. The most critical finding involved the reinstatement of encoding-specific co-firing patterns during retrieval. For each cell pair recorded from different regions, the researchers identified co-firing events during encoding for a specific stimulus. They then asked whether those same cell pairs co-fired again during retrieval when the same stimulus appeared. The repetition rate was 0.29% during co-ripple periods, but only 0.14% during duration-matched no-ripple periods, a statistically significant difference. This effect was particularly pronounced for hippocampus, cortical pairs and amygdala, cortical pairs, and held for both ipsilateral and contralateral connections. Most importantly, this repetition predicted behavior. During high-load trials with fast reaction times, co-ripple mediated pattern repetition was significantly greater 0.36% than for slow responses 0.23%. The researchers also confirmed that this reinstatement was temporally clustered around the centers of co-ripples, and that this clustering was specific to co-ripples rather than single ripples or shuffled spike controls. The author's conclusion. The authors conclude that co-occurring ripple oscillations coordinate stimulus-specific neuronal firing across distant human brain regions during working memory. Co-ripples are not merely associated with elevated excitability, they promote the reinstatement of encoding specific co-firing patterns that predict behavioral efficiency, with effects that scale with cognitive demand and extend across brain-wide distances without notable attenuation. The paper provides direct single neuron evidence for the spiking content carried during co-ripple windows, supporting a model in which co-ripple mediated coordination of unit firing serves as a fundamental mechanism for integrating distributed neural representations during human cognition.